online faculty development program organized by IQAC CPA College. Today's program is to handled by Dr. Yangovan A. So I welcome and hand over the session to resource today chairperson Professor Kumar Raja, HOD of Computer Science. Thank you very much.
Yeah, I'm very happy and invite to introduce today online faculty development program, the resource person Dr. Andy Longovan, Professor of Chemistry, Director in Charge, Center for Distance Education, formerly Head and Chairperson, School of Chemistry, Bharat University, Trichy, Madurai Kamaraj University, Madurai. And we studied MSc Chemistry from PUC College of Arts and Science, Bharat. He did his PhD in synthetic organic chemistry, Indian Institute of Chemical Technology, Osmania University, Hyderabad. Then, first he appointed as a scientific officer in Indian Institute of Chemical Technology, Hyderabad. Then, Electrochemical Research Institute, Karakudi. Then he promoted as professor for chemistry, Bharat University, Trichy. Then the position, academic administrative position, a director in Center for Distance Education, Bharat University, and professor and the former head Department of Chemistry and former chairman School of Chemistry, Bharat University. The fellowship and honors. Japan Society for the Promotion of Science, Postdoctoral Fellowship, Graduate School of Pharmaceutical Sciences, Tokyo University, Sendai, Japan. Indian National Science Academy, German Research Foundation, Bilateral Exchange Fellowship. Then a 3-month visit to Ludwig Maxwell University, Munich, German. Then President, Institution of Innovation Council, Bharat University, Trichy. Then is the member of board of studies in chemistry for 16 colleges. Then foreign countries visited for academic purpose, Germany, France, Russia, Japan, etc. Then the teaching experience for postgraduate and the PhD for nearly 18 years. For research students, the experience 32 years. The synthetic organic chemistry, especially the interested in synthetic organic chemistry, that is total synthesis, bioactive, natural products, organic catalysis, and solid phase organic synthesis, electro methods, etc. Then teaching particularly in organic chemistry, that is reaction mechanism, spectroscopy, asymmetric synthesis. He produced 11 PhD degree holders completed. Ongoing PhD are 8. Then he published the international journals 78. Then he attended the invited lectures 75, international 9, national 66. Number of workshops attend 10. Then PhD MPhil chemistry thesis evaluated 125. Today The session is drug discovery. He deliver the drug discovery for the research person Anduveli Ilangovan. Thank you, one and all.
Thank you. Are you able to see my slides?
Yes, yes. We can able to see your face and the slide also.
Thank you. First of all, I would like to thank my classmate, +2 classmate, Mr. Professor Murugesan. He is a long-standing friend. For giving me this opportunity to join in this meeting and present some of my views and then experiences in doing kind of research and other things. And I also, since I belong to this Theni district, it is also kind of a homecoming feeling for me, but it is online. You know, this COVID has made us to learn certain lot of new things in life, like which we are not used to, like isolation, self-isolation and other things, social distancing and other things. In addition to that, I would like to say this kind of online technology, we started using it. It is a kind of a blessing in disguise, I would say. And also I thank Professor Kumar Raja, the chairman of this session, for introducing me. Probably it was a long introduction, which is probably not needed. Thank you, Professor Kumar Raja, once again, my chemistry fraternity of the college. See, for today's talk, when Professor Murugesan contacted me, I thought of talking about the drugs and kind of about the vaccines from test tube to market. You know, being a COVID infected situation, and we are all facing kind of unprecedented situation, health kind of situations throughout the world, not only in India. I thought talking something related to COVID or something related to drugs, which is needed, which is a need of the hour, would be of a kind of relevant topic for this refresher course of college teachers, those who are from different kind of areas, different subject areas. But the topic is supposed to be a common topic, which is of kind of use for all of us. I would also touch upon the vaccines, the development of vaccines, how things are involved. These are all the slides which I borrowed from online. I would like to, this nowadays preparing seminars also becomes a little bit easier, because thanks to Google, whenever we search online, we get a lot of online resources, and with that it becomes easier for us to share kind of our experiences in this particular area. See, as Professor Murugesan said, or Professor Kumar Raja said, I'm a basically organic chemistry man involved in the medicinal chemistry research area. No, I know. Also, we try to make develop technology for drugs, synthesis of drugs. You know, for today's talk, I have divided this into steps involved in drugs, vaccine development, kind of hybrid organic molecules approach for the development of drugs. That is our own experience on the synthesis of hybrid organic molecules. Yeah. So looking at this slide, you will be able to see whenever there is a technological development, and population also has grown exponentially. This was the slide which I got from internet. The reliability of data is, I'm not very sure, but it looks reasonable for me to agree with this, what has been, what is appearing in this slide. You know, the population in millions, if you look at beginning of second agricultural revolution, and that was around 1500 years. Now we are in 2021. Around these days, after the invention of medicines, invention of penicillin, discovery of DNA, and then computers, all other kind of war and malaria, you can see that kind of life expectancy as well as the population growth has one way or the other has something to do with the development in the area of medicinal chemistry, or the kind of health conditions, improvement in the health conditions with the available science and technological means. Humans try to avoid death and then try to increase their life expectancy. That way, this COVID has come, probably the nature has its own way of tackling the problems. COVID is the one thing, COVID-19 is the one thing which is playing a havoc among our humans. Not only in India, it's throughout the world, it is playing a serious havoc. See, steps involved in the drugs as well as the vaccine development. That is the first part I'm going to talk about. If you look at India, is supposed to be a drug major. Next to China, are both in China as well as India, are equally competitive, equally capable of making drugs in a bulk level. Bulk drug synthesis, India is a leader. Also Chinese, we are competing with Chinese in several areas. It is worth 18 billion in 2012, 18 billion US dollars, and US dollar of 6 billion in 2005 has grown into 18 billion. More than 24,000 large and small, very small manufacturers are involved. Probably this slide was made a little earlier. Now this number might have increased actually, I believe. About 55,000, then 0.5 million kind of a chemist. It's a very huge number, over 1 billion plus people using medicines. And 90% of the domestic bulk drugs requirement is met by Indian industry, in India by Indian industry. That means bulk drug industry contributes to the 51,000 million of exports rupees, or more than that. Now, by now, it must be, definitely it might have increased. Local drugs companies, these are all the shows. See, unlike we are used to kind of a space technology, we know that India is a leading player in space technology as well as the information technology. But it is also important to know that we are global players and the leaders in the bulk drugs as well as the synthesis of some kind of a new molecule development. Also, it's picking up in India. You know, we have Bharat Biotech, then ICMR. We have demonstrated that we could also come out with vaccines at a very quick, in a very quick time, and we were able to see that we were able to, like any other country, like developed countries, we were able to develop vaccines very quickly. Then we can meet the requirement, the need of the hour, we will be able to deliver things. If you look at the drug discovery, why I want to talk this drug discovery means, we were used to kind of companies charging too much of money for drugs. For example, cancer drugs or a kind of tuberculosis or anti-malarial, those kind of unlike generic drugs. In case of specialized drugs, the companies used to charge hefty or a higher price. I think with this drug discovery process, if we can understand, we will also be able to understand the problems faced by the industry. Why these companies are interested in making a lot of money, or they are charging higher prices for their drugs. See, in a drug discovery process, there are 5 steps are involved. And now, during these 5 steps, you can find starting from a simple, not only chemist and biologist and the doctors, there involves a lot of bureaucrats, governments, and then businessmen. So many people are involved. I think running through these slides, next few slides, that will give you idea about why how these people are involved. Different kind of bureaucrats and then government and then so many other people are also involved. Before going into details, let us see what you mean by drugs. What is meant by this drug? As defined by US Food, Drug and Cosmetic Act, a drug is any substance that can be used to modify a chemical process or process in the body. For example, to treat an illness, relieve a symptom, prevention of a disease. Relieve a symptom, prevention of a disease, enhance performance. It's like improving our mood. It's kind of a changing our mood, kind of a ability, alter states of mind. All these, the chemicals involved in all this process of a body can be considered as a drug. We only consider drugs which are used to cure a disease. But we are used to kind of a definition that drugs are one which directly cures our body from a particular disease condition. Otherwise, ability to alter the states of mind, enhance performance, relieve a symptom, all these things also can be considered as a drug. Aspirin, antibiotics, caffeine, and others are some of the examples you will be able to see. See, when you look at the doctors, they generally believe that all the drugs are from the natural sources. And also, we look at the natural product, the sources which we obtained from the plant sources or the animal sources, or otherwise a kind of that has been originally this kind of inspiration or the ideas are originally derived from the natural products. That many of our doctors and many of our conventional MBBS doctors, they ignore and they several times, in fact, they try to make fun of it. I know my relative doctor, he used to say this kind of Siddha treatment, the plant-based treatments are all humbug and it has no scientific meaning. But they should understand that all these sources of drugs are from the natural sources. They are all mainly from plant sources. See, not only a drug can be a chemical or biological substance, a synthetic or non-synthetic origin. Animal insulin is a drug which used for treatment of sugar problem, this diabetic conditions. But those drugs are originally isolated from insulin is a pig or cow, and growth hormones for men, progesterone, estrogen, all those things are derived from originally obtained from humans. Then plant, like several alkaloids, like morphine or ajmalin, they are obtained from plant sources. In fact, once you make it in a large scale using in an industry, we try to ignore or forget the inspiration or the idea or where from this kind of original molecules has been obtained. That way, this considering these kind of sources of drug molecules or drugs is very important. Our understanding and appreciating the importance of the sources are very important. Synthetics, inorganic also you can say arsenic or cisplatin, a famous anti-cancer drug. In mercury, it's a drug we are using it in several traditional medicinal preparations. Like this, inorganic compounds, for example, nowadays ruthenium and ruthenium-based several drugs are coming up. Even copper-based drugs are also metal-based. Metal in organic complexes, so drugs are coming in. This synthetic is the one which is ruling now in the entire world. You can see that synthetic drugs are nowadays taken up, and it is gaining a lot of importance. Or otherwise, these synthetic drugs are originally the ideas derived from the natural sources. The earliest medicinal chemistry was from the folklore. That is the traditional, we depend upon our grandma or grandparents or our forefathers, those who developed medicinal chemistry. We must be thankful to them. Chinese emperor, we are all used to kind of Chinese medicine and Indian medicine. We all know about Siddha, Ayurveda, and other things. That is the reason I'm just mentioning about the Chinese and other kind of medicines. Chinese Emperor Shen Nung Book of Herbs. It was made before several years. Contains alkaloids used today in the treatment of malaria and for fevers. Modern therapeutics extracts of foxglove plant cited by Wells physicians in 1250 used to treat congestive heart failure in 1785 contain digitoxin and digoxin, today called digitalis. They are all originally were used for a very long time in a folklore or traditionally by our parents or grandparents. At least quarter of all the prescriptions dispensed in the US and UK contain molecules derived from plants. Look at this molecule, it's a very good number. See, at least a quarter of all the prescription dispensed in UK and US, they are containing molecules are derived from plants. 12 out of 25 highest earning in 1995, the molecules, highest earning molecules are derived from natural products. That means ethnographics shows that humans are the greatest botanical experimenters. Like this, you can see this morphine and codeine. This this in Tamil we say kanja or codeine. This abin. These are all can be used as a very good painkillers or several steroid-based drugs like this can also be obtained, or they are originally from natural sources. Clarithromycin, cyclosporin, clavulanic acid, ampicillin, this penicillin-based drug, they all are from natural sources, which we started synthesizing in bulk, and then we started forgetting it. Some of the prominent system of medicine, as we know, this is an Indian system of medicine, Ayurveda, and then Siddha, then Chinese medicine. African traditional medicines are also very famous, but many of us, we do not appreciate this kind of African medicinal traditions. Homeopathy, allopathy. This allopathy is the ruling the world now, because you can make it in a large quantity, and you can make it in a bulk using synthetic means, where really chemists, they contribute a lot for the improvement of the allopathy is backed up totally by chemists. Without the chemist, they will not be able to survive. Doctors will not be able to manage with the availability of drugs. In 1800, natural sources were only used, small scale, not purified. But in 1990s, synthetic source, unlimited possibilities, highly purified, 100% purified, one particular molecule, active principle can be prepared in bulk and used. Mechanisms partly understood, and the mechanism. See, once they have a scientific finding for one particular drug action, they claim that only these kind of synthetic medicines can be a valuable treatment method. This is a kind of what we call it as a rational drug design. Rational drug design is what? Rational drug design is a kind of approach involving computer-based design, studying about the at the cell level, and then administrating to the human at clinical trials, then computer-based modelings and other things are involved. And then with the improvement in the technology of in the biology, many new understanding about the modes of action was observed, And with this, we call it as a rational way of designing a drug. It is like a one particular disease that one particular molecule can act upon and forming a valuable drug. When a biology people alone try to do drug development, it would be something like traveling a horse and a cart separately. But when a chemist and a biology together and they put efforts, it would be something like joining together. It will make wonders, and we can quickly make wonderful stuff. Achievements or developments in the drug discovery area. See, drug development, the process involves 5 steps, I would say, 5 important steps in drug discovery. You have to start from, you have to start from a person who is identifying the problem. See, the person who is identifying the problem, then chemical has been designed using computer modeling, then it goes to the animal level, then it goes to the chemical synthesis, then followed by which you go for a cell testing, and then human testing, clinical trials, FDA approval, followed by which it goes to the final market. It becomes available in the market. Identification of a useful disease target, identification of a lead molecule that works in the laboratory, choosing the bioassay, clinical studies, obtain the test, step 4, test of humans, that is the phase trials. That is what is missing really now. In case of a Covaxin or any kind of a vaccines available for a COVID, now clinical trials are really missing. How much time and what are all the steps, what are the intricacies involved in this kind of every step? Let us see one by one in detail. Who is going to identify identification of a useful drug disease target? See, for India, tuberculosis or cancer or kind of diabetes is a very serious problem. But in case of developed countries like US, obesity, psychological problems, and other problems, like for us, malaria is a very serious problem. But a Western country, malaria cannot be a problem because they live in a cold country. Like this, identification of a useful disease target, and which target I'm going to develop a drug, which target I'm going to make this drug available in the market, that is being identified by kind of a corporates, decided by corporate companies and government of India. You look at this is a kind of a different type of drug disease conditions. Oncology based drugs 18%, and diabetes 24%, neuro disease kind of a 12%, virology contributes to 6%, infectious disease by bacteria, immunosuppressive diseases like this. So many, this is something like in 2009 till 2009 it was like this. On 2010 you could see the situation changes. Then the other diseases has taken over, diabetes level and other things. Like this, depending upon the time and kind of a need, the corporates, those who are involved in making money, they decide which drug molecule, which drug has to be developed, what kind of disease has to be addressed, what kind of the identification of lead molecules that works in the laboratory. This is where, as I said, in this case, corporate companies and government officials, those neglected disease, like for example, in our case, tuberculosis, not many companies may be interested, or kind of a malaria, many foreign companies may not be interested, but the government of India, in order to avoid problem faced by us, government of India, that means will be involved, and then corporate companies and the government of India, that means bureaucrats and the politicians, they're all involved in the drug development. You should understand that. Please understand that this government and then corporates, like politicians, starting from politicians, everybody is involved. Identification of a lead molecule that works in the laboratory. Natural products, medicinal fork, combinatorial synthesis, computer design. All those things are done by chemists, and the computer-based people like Professor Murugesan, who is doing a computer subject, there also can be involved. And receptor, enzyme, nucleic acids, serotonin receptors, migraine, dopamine receptor, antidepressant, fluoxetine, many targets, but few molecules are available. Now, depending upon the how many number of molecules are available for each target, we try to identify or work in this particular area where the number of molecules are less. I'm showing you an example of azithromycin, which was the drug which was developed for anti-HIV treatment. This azithromycin works on the principle of chain termination. Look at this. This is the ribose. If you have OH group at this place, azide group, and then instead of azide, you have an OH group, one more OH here, it will be like a ribose. That is the ribose sugar, which is part of our DNA. You can always find a ribose sugar. But what they did was they designed a molecule. Instead of a OH group here, they introduced azido group, N3. That azide group, once introduced, what is happening? It will behave something very similar to a DNA material, that is thymidine. Adenine, guanine, thymine, cytosine. Thymine. This is a thymine-based molecule, which is a part of our DNA, growing DNA of virus. This DNA virus, thymine, cytosine, adenine, again thymine has to add. At the time, this molecule will go, which is a drug molecule. Azithromycin is a drug which we administer for treatment of HIV. That goes and it mimics, try to mimic like a thymine. That is thymine containing ribose sugar. Instead of ribose sugar, you have, instead of having two hydroxyl groups here, you have azido group. That means unless there is a hydroxyl group, that the chain elongation, you have to go on adding like this continuously. It has to go on elongate. It has to extend it. Then only the virus can survive. If we can stop this kind of a elongation of the virus DNA, viral DNA, you can stop the life cycle of DNA virus, life cycle of DNA generation, and then life cycle of the virus. That is the original idea. Like this, you have to, depending upon the mechanism of action, sorry, how this life cycle of enzymes, life cycle of particular virus or the bacteria, based on that, if you can rationally design some molecules which is very closely related in structure, then we will be able to develop drugs which is of useful for treatment of a particular disease. Like this, here you can find the chemists are involved, a lot of chemists are involved. Here you can see drug design. It involves the synthesis, high throughput screening, that is again chemists are involved, X-ray crystallography, again enzymes are, chemists are involved. You can design the molecules through molecular modeling, molecular ligand design. It is an enzyme model on which we are trying to fit in the molecule, or whether how best it can fit in. Accordingly, best fit molecule can be synthesized by chemist, then computer-based design, then synthesis, then high throughput screening by chemist and biologist, structure activity relationship study. Then structure activity relations study gives us an idea which functional groups has to be introduced. Again, chemists are involved and biologists are involved. Here you can find that botanists, those who can develop or grow a particular plant, they can also be involved. And then comes the synthesis. Synthesis, synthetic approaches, preclinical research, and then rational drug design, all those things. You can make so much of molecules by combinatorial synthesis, or depending upon this is a DNA target. This is an enzyme target, enzyme-based target. This is again, I would say enzyme-based target or a DNA-based target. You can make so many molecules in the laboratory but only few molecules are found to be active. You cannot let us say if I'm synthesizing, able to synthesize 1,000 molecules at a given time, I would be able to find only one among the 1,000 molecule may be showing a proper biological activity. Like this, you can see complex disease target. Most compounds do not become medicine. Like a filtering, through the process of filtering, only few molecules comes down, and many molecules goes out of the trial, and they'll be neglected. Here comes the choosing the bioassay. Test the lead molecule in the lab and on animals. First, we all know, many of us, we know that isolated cells, tissues, enzymes are receptors. Enzyme models are enzymes. For example, in case of a candida, phospholipase is the enzyme. That is the virulent factor or the active principle for the the candida albicans. That is the fungus. It is a very commonly found in people undergoing operation. Means that immunosuppressive action like candida can also be a lethal. For candida to act, this phospholipase is the enzyme involved. But in order to find a drug against candida, that is the fungus, you have to see whether your molecule is active against particular enzyme, that is phospholipase. Like this, enzyme models also can be used, tissues, and then isolated cells, cell lines, HeLa cell lines in case of a cancer. Like this, receptor models, many other things, clone and express in the, can be used as the models. And then if you find this kind of a interesting activity by one particular compound, then you identify, take that compound as a lead compound and develop it further. Yeah, that is the idea. Clone and express the fast dividing cells such as bacteria, yeast, and tumor cells. Here biologists are involved, so much of biology, microbiologists, and then biochemists, and then I would say then biotechnologists, many people are involved. Different steps involved in biological studies can be said as a drug taken orally reaches, how it reaches a target, how it reaches within the body, where it goes, what happens to drug once inside the body, routes of administration, external administration, parenteral or trophical or inhalation, phonophoresis, intranasal, entophoresis, instillation. So many different ways you can administer a drug, or how these drugs are being absorbed in the intestine or by the skin. That has to be studied by biologists. Test of bioavailability. This is the fraction of administered dose that reaches the systemic circulation. Bioavailability means you may consume 1 gram of a drug, but once it goes to the system, only 500 milligrams will be available. Then 500 milligrams will go into excreta, it will go as a waste. Bioavailability, whether it has to be checked. Pharmacokinetics by route of administration. Metabolism, how these drugs, once it goes inside the body, whether they retain the structure or it undergoes some kind of a chemical changes. For example, under the biological pH, a ester group may undergo hydrolysis, an amide group may undergo hydrolysis. Pharmacodynamic studies. Like this, so much of studies are involved, in biologists are involved. Like in vivo testing the animal, transgenic animal, induce in vitro is like a testing on the cell lines. In vivo is testing on the model, which is like a monkey or a mice or like a testing on the transgenic animals like a mice. You have to test. That is the next level. Animal testing. Contribution by microbiologists and animal scientists also. Now comes the medical doctor. In case of drugs, the it is like a doctors are equivalent to heroes in a cinema, and the heroes and the heroines. And the director may be directing, or so many that technicians are involved in lighting, and then we call the so many things are there in a cinema making like this. Many people are involved, but the credit goes to the doctors, those who are prescribing the drugs. Here comes the doctors. Here comes the clinical studies, test on human. Here comes the phase 1, phase 2, phase 3. What is happening in the phase 1? Introduction of investigational new drug to humans. The step at the step 4 only we involve it comes to humans. And then healthy volunteers, subjects, patients are identified. The evaluation of drug metabolism, again, what is happening in our human body, structure activity relationship, mechanism of action, side effects, and increasing doses, pharmacokinetics and pharmacological effects. Typically, and then once initially you test on a few number of volunteers, like let us say less than 100 volunteers. Once you are happy or finding it okay with these less than 100 volunteers, then you go to a larger number of patients. That takes time. Phase 2, effectiveness on patients with particular disease. Volunteers may not have a disease or may have a disease, or here in this case, phase 2, we identify particular disease condition, the person typically well controlled and conducted a relatively small number, usually involving several hundred people. This is only less than 100, this is several hundred, expanded, controlled and uncontrolled trials, overall benefit risk relationship. Like this, you can exploiting the results of general population, physician labeling, then usually include several hundreds of thousands of people, decided by physicians, regulatory authorities. This Food Drug Administration authorities I was talking about, this is the phase 4, they come, regulatory authorities like administrative people. Clinical trials, in nutshell, you will be able to see that approved protocol by biologists, and then investigator selection, that means the volunteer selection and approval process, then patient recruitment, then you have to monitor this statistical analysis, and everything goes on, presentation to the government and other things. It can go on like this. Phase 1, phase 2, phase 3, phase 4. 4 different phases are involved before a molecule goes into drug. Finally comes the FDA approval. Even after acquiring so much of data and so much of information, the government comes into picture. The government, bureaucrats, and social issues. Abortion is a problem in several countries, and then kind of a treatment for certain diseases are not allowed, banned in several countries, like consuming alkaloids kind of drugs is banned in Arab countries. Like this, they can be used for kind of unwanted activities. Like this, several drugs, again, controls comes by government, bureaucrats, and the social issues are also there. Food Drug Administration approval is the common approval. It is there in case of an America. This is a Food Drug Administration approval is needed. Center for Drug Evaluation Research, Center for Biologics Evaluation Research, Center for Devices and Radiational Health. So many different type of veterinary medicine. Like this is a particular Food Drug Administration is the one which takes care of approval in America, United States of America. Let us see what are the kind of administrative people available in case of India also. Drug approval process in India is taken care by Government of India, Ministry of Health and Family Welfare. The Drugs and Cosmetic Acts 1940, followed by amended in 2005, drugs and cosmetic rules. Cosmetics and drugs, they classified into two different and amended once again in 2005. These are all the rules, and then these are all the departments which takes care of availing and this awarding kind of approval to drugs in India. They are like drugs Directorate of General of Health Services, Central Drugs Standard Control Organization. They are available in different parts: Kolkata, Ghaziabad, Mumbai, Chennai, and then Guwahati. So many places they are available. Headquarters, zonal offices, sub-zonal offices. You know, you can think about when one has to go through so much of processes, you can think about how much of administrative formalities are involved. Now, I would like to emphasize that in order for a drug to start from a test tube, let us say at this level, in order to go from here to here, preclinical research, clinical studies, FDA, NDA review process, that the bureaucrats will be reviewing what are the outcome of the clinical studies. Like this review decision, so many things are involved. This much time, it is almost like a 13 years, 10 to 13 years maybe involved in drug development. Long time and huge money is involved. People say for a drug to be developed, it involves like for a single drug to be developed, it is like 1,300 crores of money for a molecule to start from zero clinical studies to here. Sometimes many molecules post-approval study, ND approval reviewing, phase 1, phase 2, phase 3. Phase 1, phase 2, phase 3, it will be some 12 years will be over. Finally, it takes 18 years for in almost all countries, this is the common process. That is the reason many drug companies are charging heavily when they introduce a new molecule into the market. They invest a lot of money, involve so many people in the development of drugs, and the time is also time, money, manpower, so much of things are involved in drug development. You know, we know the famous example of accidental discovery or serendipity. Accidental drug discovery or lucky way of to discover drugs. Serendipity, we in a very technical term, we call it as serendipity means happy accident. Like this, here a man trying to mix up so many chemicals and ends up with a molecule which is found to be a very useful activity. Like accidentally he discovers it. Serendipitous drug discovery has played a very famous, very useful role. Nitrous oxide ether as a narcotic gas in surgery resulted from observation that the people who inhaled these chemicals did not experience any pain after injury. Nitrous oxide as a narcotic, it was used as a narcotic. It was found to be useful for analgesic kind of the vasodilatory activity of amyl nitrate. These are all the earliest examples. Let us see what are the recent examples. Aspirin, acetyl salicylic acid, was thought to be just a better tolerable prodrug of salicylic acid, but turned out to have a unique mechanism. You know, nowadays aspirin is used for dilating the blood clots and so many headache. Aspirin is the useful drugs during those childhood days. We used to go for whenever people get a headache, people used to go for an aspirin. Like aspirin nowadays, it is being used in as a bit of a kind of heart-related diseases. Even COVID-related disease, aspirin is found to be useful. Phenolphthalein was considered as a useful dye for cheap wines. When phenolphthalein was mixed, it was found that it can act as create a diarrhea. Like this, Viagra was originally designed as a beta blocker. That means for heart disease it was designed, but it has become a blockbuster or famous drug for impotency, to treat impotency. Isoniazid, like this, so many. Vimblastin, vincristin is a Nithya Kalyani நம்ம எல்லாருக்கும் தெரிஞ்ச ஒரு பூ தான். நம்ம நித்திய கல்யாணின்னு வீட்டு முன்னாடி வளரக்கூடிய ஒரு செடி. அதுல இருந்து கிடைக்கக்கூடிய விம்பிளாஸ்டின், வின்கிரிஸ்டின் 2 alkaloids. They can originally prepared to test as a drug for anti-epileptic. கை வலிப்பு வர்ற வியாதிக்காக டெஸ்ட் பண்றதுக்காக ஐசோலேட் பண்ண இந்த ட்ரக் வந்து found to be a very famous anti-cancer agent. It becomes a famous anti-cancer agent. See, being we are involved in I'm having another 15 minutes to go, I think, or 11 minutes to go. Let me complete with this vaccines. COVID vaccines, a race against time in the middle of death and devastation. This is the topic title, like I saw during search for some of the slides. A race against time. We were talking about 18 years or 12 years or phase 1, phase 2, so many clinical trials. When people are dying and this kind of a FDA approval and so many involvement of so many other people in it happens in quick succession. It happens very quickly. People parallelly work on it, and then several of these rules are diluted, and the process of approval and reaching to the treatment of the people who are in need, it happens quickly. How it happens, let us see. In case of a Covaxin, we know the Covaxin is an Indian based vaccine available. Vaccines are, what are the vaccines? Basically, vaccines train immune system to recognize dangerous pathogen. Whenever there is a bacteria or fungus or a virus comes inside, our immune system try to identify this foreign invading organisms and fight the infection without having to get sick, without நமக்கு எந்த விதமான ஒரு சைடு எஃபெக்டும் தராம, சைடு எஃபெக்டும் ஒரு நோய் சம்பந்தமான சூழ்நிலையை ஏற்படுத்தாம நம்மளுடைய இம்யூன் சிஸ்டத்துல இருக்கக்கூடிய this kind of molecules like T lymphocytes and then B cells, they fight, and then these are all the vaccines molecules. They are naturally available in our body, B cells and then T cells. இந்த B cells-ஐயும் T cells-ஐயும் குயிக்கா ஆக்டிவேட் பண்றதுக்காக ஒரு சிஸ்டம், ஒரு மெடிசன், ஒரு கைண்ட் ஆஃப் மாலிகுல்ஸ் injected into your bodies are nothing but vaccines. Train the immune system. How does this happen? When a pathogen infects the body, the immune system dispatches an army of different cells to clear the infection from the body. 2 types of immune cells that make up this army: B cells and T cells. B cells and T cells move through the body constantly, regularly. They are like a policeman. They'll be always moving throughout the body. You know, we were knowing about the blood circulation, but unlike blood circulation, there is another blood and lymphatic fluid. Lymphatic fluids, they are available in the joints. Then whenever B cells bind to antigens, they can travel and check through the different places like a policeman, whether there is any invading external organisms comes inside the body. These B cells and T cells, they go and bind to antigens. They crank out thousands of antibodies. They generate antibodies, which in turn bind to the other pathogens circulating the blood and lymphatic fluid. நம்ம ரத்த ஓட்டம் மாதிரி நிணநீர் ஓட்டம்னு சொல்லக்கூடிய lymphatic fluids are also important. In order for you to activate this lymphatic fluid, இந்த நிணநீர் ஓட்டத்தை நீங்க சீராக வைத்திருப்பதற்கு, you have to do kind of a physical activity. Blood circulation is taken care by the kind of your heart, but in case you want to activate your lymphatic fluid, you have to do a physical activity, physical exercise, without which it cannot be activated. These antibodies send chemical signals immune cells to help and destroy the pathogen. In the vaccine, that train the immune system, B cells and T cells, that can train against a particular invading organisms. How are the vaccines approved in the United States? I'm taking an example of United States again. It begins with preclinical research. The vaccine is first tested. Again, the same story, like drug discovery, அதே story தான் continue ஆகுது. If a vaccine passes through preclinical stage, it begins 3-phase trials. First phase trial between 100 and this is between 100 and then hundreds of volunteers in the phase 2. Thousands of or even tens of thousands of volunteers receive their vaccine or placebo. Placebo is nothing but without having a medicine, something like you are injecting a distilled water or no medicine, but it will have a psychological effect. Amid COVID-19, volunteers are encouraged to social distance, wear masks, and follow their public health infection prevention guidelines. Researchers will track how many volunteers get COVID-19. For COVID-19 injection vaccine develop பண்ணதுல, in order to check that, they have to go through all this process, phase 1, phase 2, phase 3, and then researchers will track how many volunteers in the test எல்லாத்துக்கும் நடக்கிறதுக்கு தான் several years ஆகும். We know this COVID has started in 2019 December. Now it is almost less than 2 years, and we are already having a vaccine. That means the vaccine has come out of very kind of a quick approval process. Researchers will track how many. After all the 3 phases, if the vaccine is found to be safe and effective, then it is submitted to FDA for approval. A multidisciplinary team of scientists reviews the data collected throughout the trial. Throughout the trial, lot of data are collected. The data log look good. The FDA will license the vaccine and manufacturing process. This is supposed to be the originally the idea of doing this. Even after the approval of vaccine, FDA and the Center for Disease Control Prevention continues to monitor its safety in all the process. But experts like see, and it has to report after effects, whether is there any side effects. Let us say a person taking a vaccine, if he loses his life, it is very dangerous. Like this, people say this is also happening, but it is not scientifically proved. In all this process, vaccine development from preclinical to license and the vaccine production usually takes a decade or longer. Decade means 10 years, almost more than 10 years. The fastest vaccine ever successfully developed by mumps vaccine produced by Merck in 1919 still required 4 years. Minimum 4 years was the previous history. Our previous history says that in case of a mumps vaccine develop பண்ணதுல கூட minimum 4 வருஷம் எடுத்துட்டு இருக்காங்க. ஆனா coronavirus COVID vaccine just we have taken less than 2 years. Experts like Yoaki do not see this as a cause of alarm. The vaccine candidates undergo rigorous testing before being approved by FDA. Even though the timeline is rushed to develop COVID vaccines, the evaluation of safety and efficacy is not being compromised. People claim that you look at this is an interesting table I want to show you. இது வரைக்கும் நம்மள்ட்ட உலகம் முழுக்க 10 different kind of vaccines available இருக்கு. Comirnaty, Modernaவுடைய COVID-19 vaccine, AstraZeneca, that is Covishield, Sputnik னு சொல்லக்கூடியது, this is from Russia, Johnson, that is from Johnson and Johnson, and then this is again Sinovac, this is Chinese based. Chinese, they have made 2 different vaccines. Chinese, this is the VBB-QRV, this Chinese, they have made 2 vaccines. Netherlands, they made 1, Russia, UK, US, and then India, and then again 1 more Chinese, and then ours is the Covaxin. So many vaccines are now available. These vaccines are, one is based on mRNA based. This is the kind of supposed to be a very revolutionary technology. None of the virus till today available in the market for any other kind of a disease are based on mRNA. This is the kind of a new technology which they introduced in case of coronavirus. Adenovirus vaccine, that is inactivated vaccine. This is ours is also inactivated vaccine. Covishield, this is a Covishield. In case of a Covishield, they are using a kind of coronavirus isolated from chimpanzee. They modify the outer shells, and then spike proteins they introduced. Then with that they introduced to the humans. That is a modified virus. virus particle otherwise. In case of a Covaxin, this is nothing but this is thermally isolate the originally isolate the kind of coronavirus and then kill it thermally without disturbing or destroying the outer shell of the vaccine, outer shell of the coronavirus. It is called of a killed virus, inactivated vaccine. Inactivated vaccine, like this inactivated vaccine based are available in China, in India, like this the inactivated vaccines are available. Covaxin is a vaccine which is based on the killed virus, or like a it is like a original shape will be there, but once body it gets into the body after the injection, the body cells they will recognize this as originally as an invading virus. ஒரு வைரஸ் உள்ள வருதுன்றதுக்காக அதை ஐடென்டிஃபை பண்ணுவாங்க, but it is not actually live virus. It is a killed virus. Thermally it was heated and it was killed originally. Inside it was killed, outside it was the same design, shape is retained. See, usually classical vaccines in any kinds of mumps or polio, many other vaccines we know, classical vaccines preclinical stage 18 to 30 months, and but in case of a Covaxin only 0 months. Phase 1 6 months only taken, but usually it used to be 30 months, but it in this case 6 months. But phase 2 க்கு பாத்தீங்கன்னா 32 மாசமாவது ஆயிருக்கணும், பட் இதுல 6 மாசம் தான் எடுத்திருக்காங்க. இதுல பாத்தீங்கன்னா 6 6 6 3 6 18, this is 12 to 18 months, less than 1, it is like 1 to 1.5 years was involved, but in this case usually it takes 9 to 12 years approval, phase 1 phase 2 phase 3, like this. In case of a Covaxin, lot of complications are involved. Logistics chain for COVID vaccines, logistics main effectiveness of vaccine, this is like a minus 20 degree, which is the Pfizer biotech based vaccines has to be stored at minus 20 degree. This is, you have to throw the this is Moderna, I'm sorry, this is Pfizer is minus 70, Moderna is minus 20. minus 20 ல மெயின்டைன் பண்ணணும். Pfizer வேக்சினை யூஸ் பண்ணணும்னா அது ப்ரிப்பேர் பண்ண இடத்துல இருந்து from the place of manufacturing it has to move to a different place where people started using, it has to maintain, the cold chain has to be maintained. That is the biggest challenge. One more challenge in vaccine development. I think I'm very close to the time. Shall I take another 5 minutes more? Professor Murugesan?
Yes, sir. Yes, sir. Yes, sir. You can proceed. No problem.
Please. Okay. COVID vaccine courses, the estimated remaining vaccine courses, Moderna, so much of AstraZeneca. This is some of the vaccines which is very commonly, widely used. AstraZeneca, Novavax. I told you some 2, 4, 6, 10. These 10 to 11 different vaccines are available as of today. The estimated remaining vaccine courses, reserved courses, these are all the availability. I'm talking about the availability. Who funded the COVID vaccines? You look at this private parties, lot of investment by private party, not for profit organizations, something like a philanthropist. Government is involving very less. You see this blue color, Moderna is totally involved by government. In case of Novavax or AstraZeneca, lot of private parties are involved. In case of Sino, these private parties are involved. Sanofi, private parties are involved. GSK, Glasgow, SmithKline Beecham. Like this, not only who has funded the COVID vaccines, different vaccines are taken care by not only by government, non-profit making organizations, private parties, so many people are involved. Here you can see this phase cost per is, this is preclinical phase, cost is the 165 million. Phase cost is 289 millions. Like this, high cost and is involved in once again, like a drug molecule, the cost involved in clinical trials are phase 1, phase 2, phase 3, they are actually happening parallelly. It is being carried out very parallelly. And we started, although we started using it, this is the approval process and then clinical trials, monitoring, applying on the humans, testing on the humans. All those things are very, very parallelly it is happening and it is being monitored. We must know that it is being continuously being monitored and updated. Vaccine makers are charging different prices. Moderna is the highest priced vaccine, 25 to 35, $37. Then Sputnik is supposed to be the cheapest vaccine available in the market. Johnson and Johnson is also equally good. CureVac, Novavax, Oxford AstraZeneca. This is AstraZeneca is also reasonably at a cheaper price. BioNTech and then Sanofi. Of course, we in India we make Covaxin that we sell it at the for Indians. Actually, exploratory research 2 to 4 years, preclinical 1 to 2 years, phase 1 to phase 3 5 to 7 years. Manufacturing, marketing, it is at the manufacturing approval, all those things. It takes phase 4. It involves here, you can see overall more than 1 billion of money is involved. 1 billion of money cost. The cost is 20 million here. This is 15 million. This is here 24 million, 87 million. If you put all those things, the vaccine development process, including the number of years, it takes so much of money, Of course, now this we are making use of it in bulk. We are making use of large consumption of a vaccine is available now. That makes easy for the companies to make profit at the very quickly. That is the one way of making vaccine. I think with this, my friends, the teachers, those who are participating in this refresher course will be able to understand and appreciate, I believe, the kind of intricacies involved in synthesis of a drug or a vaccine. Not only simply biologists and chemists and doctors are involved. Beyond biologists and chemists, doctors, so many other bureaucrats, then government, and then the business person, even a decision-making process, politicians, so many people are involved in this drug development process. And it involves a lot of time, money, and then a kind of not only time, money, it involves energy, so many other things are all involved. That is the reason whenever a drug is introduced in the market, it is immediately the company try to make money, or otherwise there are like a patenting regulations, all those things. A patent validity is for 15 years only. Patent that a product patent, process patent, 2 different patents are available. Process patent is you can hold it for very few years, but product patent you can hold it for more than 10 years. Like this, companies, they try to protect their intellectual properties. Like this, so many things are happening. Actually, now it is already 11:37. I think my time is up, and I have so many other things to go. This is like how do we design the hybrid organic molecules? Hybrid of Lord Ganesh is hybrid of men and elephant. Like this, hybrid organic molecules also can be try to follow in our research group, and we have also synthesized anti-HIV drugs based on this HIV. So much of drug molecules has been synthesized, and we have demonstrated found useful activity also. This we have checked its anti-cancer activity. Okay, thank you the audience for participation. Thank you all. Thank you the organizers for providing me this opportunity. Now I would like to invite questions if there are any.
Question. Anyway. Thank you, sir. Participants, if you have any doubt, you go through chat box.
Yeah, I would like to thank some of my PhD students and this postdoctoral fellows, current PhD students. Yeah, now he's in Israel and he is in Bangalore. Three of them are working in a Bangalore company. Now he's in Hyderabad, postdoctoral student. Some of them are MSc students. I thank the funding agencies for providing me opportunity. My institution, Bharati Dasan Universities, and the UGC as well as DRDO, CSIR for providing me opportunity. If there are if there are any questions, I'm willing to accept it. Otherwise, I thank once again the organizers.
Thank you,
Thank you,
sir.
sir.
Your
Your explanation is very
nice.
nice.
So
So
far,
far, we have observed
and
to
know
know the
technology
technology
discovery. The title is very interesting and easily understandable. I hope our faculties are know about the drug discovery. Thank you very much, Professor Ilangovan. Thank you, participants also. Thank you, one and all. Thank you.
Professor, thank you very much.
Thank you, Dr. Murugesan.
Thank
Thank
you.
you.
Okay, okay, okay.